looks like it's 1:55. I guess this thing is on. Let's get started. Today we're going to try to get through completely transcriptional termination and make substantial headway on the next one, which is the operon, because we have a lot to cover for the exam, and my objective is to have everything listed in the syllabus for exam 3 to be actually on exam 3. So will I get a chance to talk about it in class? Depends on your time reference. I have talked about these things in class. I have audio files on these topics. So you're responsible to get the material from lectures, but it may not be this year's lecture. Let's just see how far we go. And I'll be giving a review session. I'll try to get it streaming and try to remember to record it this Saturday from 1 to 3 again, as we like to do on the Saturday before the last day of exams. So it opens up Saturday morning, 8 o'clock. I'll be spending Friday trying to put together question groups, and it closes down Monday night. Actually, it probably says Tuesday to accommodate people on the West Coast. So Tuesday at 3 a.m. or something like that, I think, is officially the time. External cameras will be required this time, USB external camera, unless you sign up and take it live in person, which for me is not a good option because I have to stay there all night. But you'll notice under the persons or people tab, there are groups. Again, there's a place to sign up for the 6 sessions that will begin at 6 p.m. in the microbiology computer lab. I think the last one starts at 8:30 p.m. Monday night. Okay, termination. How do you bring transcription to an end? There are 2 general strategies, but they both have something in common. Both strategies involve destabilization of that little RNA-DNA duplex that's sitting in the middle of the active site. The intrinsic method, which is the first one, factor independent, does this simply by putting the polymerase over a very unstable region and then causing it to pause. The factor-dependent mechanism involves a helicase which chases the RNA polymerase, which pauses coyly at a hairpin, waiting for the Rho factor to come up and just ratchet out the transcript from the active site, disrupting again the RNA-DNA duplex. A hairpin is involved in both mechanisms to cause the RNA polymerase to pause. Every time you see a hairpin that's made by the transcript, RNA polymerase pauses. The vast majority of the time, no termination occurs. So let's see how nature has designed this termination. We'll talk about the factor independent or intrinsic termination first. Requires a hairpin in the RNA, and there's a stretch of U's in the RNA. And what codes for U on the template strand? Since U is like T, A's have to be on the DNA template strand. That's a favorite question I ask. A series of A's will result in a series of U's in the RNA, and this U-A pairing is one of the most unstable. If you get it just to sit there long enough, ambient temperature will result in the duplex melting. If you go over it real fast, there's not enough time for it to melt apart. But if you cause the polymerase to deliberately pause while it's sitting on a stretch of A's, then termination will occur. So here you see the hairpin. Notice it doesn't have to be a perfect hairpin, and these come in all sorts of varieties, but notice in terms of configuration, the U's, which are encoded by A's on the DNA, the U's are immediately adjacent or very close to the hairpin. If they're U's right over here, it wouldn't work. If the U's are back here, it's not going to work. The U's have to immediately follow the hairpin because these U's are still in the active site of the enzyme. This hairpin has exited the RNA exit pore and has made contact with the outside of the polymerase, causing it to pause at that site. And just temperature causes this to melt. Sometimes I give you sequences on homework and sometimes exams and say, what kind of termination site is this? Of course, there are two kinds. Intrinsic, which we just talked about, and factor dependent. Both of those require a hairpin, right? So I may give you a sequence that has a hairpin and you should not go, aha. You should go, there's the hairpin. Does it have a stretch of U's right after it? If it doesn't, it must be factor dependent, a row-based terminator. If there are a stretch of U's, or in this case, because this is DNA, on the bottom strand, of course, which is the template strand, you're going to see the stretch of A's. So is this a hairpin? Well, most of you can probably do this, but what you're looking for just at 30,000 feet is, do you have complementarity? A's over here, T's over there. If this thing folded up on itself, the A's could pair with the T's. The CC, the GG, you can see that this forms a hairpin. What I like to do is just draw little arcs. I draw an arc from this block to that, and then I draw a block from that to that, and I notice that I can form sort of a rainbow configuration. And I know that's a hairpin. The next thing I'm looking at is, does it have a stretch of A's on the bottom strand? Doesn't count if the A's are on the top strand, right? Because an RNA A pairing with a DNA T is perfectly fine. It's when deoxy A pairs with the U's in the RNA, since this is double-stranded DNA, you're not going to see U's there, but this A's on the bottom or template strand paired with the U's in the RNA. That's the key to it. That's that deliberately unstable duplex that is there waiting to be melted. Of course, the RNA looks exactly like the top strand. It's just, it has U's where there are T's. Here you see the hairpin protruding from the exit pore, causing the pause. And here's 50 ways to portray termination. Let's see what this is. True or false? The mechanism of intrinsic terminators only relies on the DNA sequence. There's no secondary structure involved any place, right? Is that true or false? False, because of course you do have to have secondary structure in the RNA, in the transcript, to facilitate the pause, of course. Okay, that's intrinsic termination. That is how most termination happens, probably on the planet. Eukaryotes don't apparently terminate that way. We still don't know quite what makes that happen. It's not as precise. You get an add poly A signal, and then you get a deliberate cleavage of the transcript in a eukaryote, the polymerase continues on, but at that cleavage site, another enzyme in the complex now adds a couple of hundred A's on the end of something destined to be a messenger. So this is a bacterial mechanism primarily. Factor dependent requires something in addition to just the transcript and the DNA template. In the one case that we're going to study out of several that are known in E. coli, we're going to only look at rho, the Greek letter rho. How does it work? Rho is a helicase. That means it crawls along one base at a time. It's called a helicase, but it's related to a helicase, but most of the time it's not melting anything. A helicase crawls along single-stranded DNA or RNA in this case. This is an RNA crawling helicase. Crawls along one base at a time until it catches up with the RNA polymerase. When it catches up to the RNA polymerase, it can't go forward. It goes up against the wall of the polymerase, but continues to yank the RNA out of the active site. How does it get started? What do you need to invoke this RNA helicase called RUT? Well, you need a place for it to bind the transcript. And then you need a hairpin to be formed in the RNA to cause the RNA polymerase to pause and wait for RUT to catch up. So it's a two-component, just like intrinsic terminators are. This is also a two-component termination signal. A RUT site, which is where Rho binds the RNA, and then that's followed by a hairpin later on. So what does a RUT site look like? We'll have this in just a moment, I presume. It moves along, releases from the structure. This is all, okay. And in an intrinsic terminator, where are the string of A's? On the template strand. Okay, back to the subject at hand. What does a RUT site look like? This was very hard to figure out. Many graduate students crashed and burned on this. It turns out there is no sequence that says, hello, I'm a RUT site. It's only rich in C's. Rich in C's, that's enough to attract Rho. Also, look how low it is in G's. Rich in C's, very low in G. So that's one requirement, that you have an area that's just mostly C's. And you don't want to have any secondary structure, any hairpins that involve those C's. You want that to be in a non-hairpinned region, so it's open. So no hairpin, the C's can't be part of an inverted repeat with some G's to form a hairpin. It has to be open and exposed. Speaking of exposed, its major form of regulation is based on how exposed it is, how accessible it is. Secondary structure is important, but the next important thing is ribosome traffic. If the ribosomes are zooming over this transcript at 45 bases per minute, translating, then it doesn't leave Rho an opportunity to bind to that RNA. So you're going, well, why should a ribosome be zooming past the RUT site? I thought this was a terminator. Shouldn't terminators be placed way after coding regions? Yes, they should be. Intrinsic terminators are way at the end of the message so it doesn't interfere with translation. In fact, Rho or factor dependent terminators are way at the end of all the action, all the translation also. But we're going to learn on occasion you'll find a row-dependent terminator sitting right in the middle of a region that should be encoding a protein. That's okay as long as the ribosomes keep passing over that region, because that prevents the terminator from actually terminating, because row cannot bind to the rut site because of all that ribosome traffic. So why would nature do that? Why would it put this cryptic terminator right in the middle of something that's as important as a coding sequence? Is that deliberate, or are these just there by serendipity? Well, it turns out it provides a means to regulate. This is a way that the cell can test the waters and say, how active am I? Am I in a dormant state? Am I in starvation mode? Have I run out of substrate? Then I shouldn't be translating all these enzymes to degrade all these non-existent substrates. This is a way the cell monitors its physiological state by having a process, transcriptional termination, dependent on translational activity. Could you recognize? I hope so. Rut sites rich in C's, poor in G's. No secondary structure. The more C's you have, the more efficient it is. This is what the rut site, what row, looks like. It's like a typical helicase, 6-membered ring. It must move faster than RNA polymerase to catch up, or the polymerase has to be paused at a hairpin. In which direction does it move? You can just sort of figure that out. Since the transcript, the 5' end of the transcript is the first to come out, row is going to be moving from 5' towards the 3' end. So back to this ribosome deal, because this is going to be an exam question. I'm going to put a diagram that I'm going to show you. I'm going to show you the exam question and tell you how to think about it and explain it. Not a good time for a nap. This means no ribosome should occur between it and the RNA polymerase. That's another important point. If row binds at the rut site and it's trying to catch up with the RNA polymerase, then you don't want a ribosome to be in between you and the enzyme. Now, is there anything to prevent a ribosome from being in between between row and the target? Can ribosomes just bind anywhere, or do they have to bind at the 5' end and also work their way along translating? In a bacteria, that's true, they have to do that. But in a prokaryote, a ribosome, any stretch of RNA is fair game. It can bind right in the middle of a polycistronic transcript and just start scanning, looking for a Shine-Delgarno sequence. So again, if your ribosome traffic is too high, the row-dependent terminator is not going to be very efficient. Also, if you have the hairpin a long way away from rut, you also reduce the efficiency because you increase the chances that a ribosome may intervene in there. A row review only works if RNA is not being translated, ribosomes are not in the way, provides a link between translation and transcription. Row is a helicase, a RNA-dependent ATPase. It only cleaves ATP if RNA is present. It needs it to move one base at a time. It will only unwind a double helix if it's a RNA-DNA helix, which is called a duplex. It doesn't unwind DNA. Except I think the mechanism of unwinding is unlike any other helicase, because how can Rho crawl inside the RNA polymerase to melt the hybrid? Ridiculous. The way that I envision it, which you won't see in any of the journal articles, so it's probably totally made up and false, is that the RNA comes up and pulls it out, burning ATP to do that. Just pulls the transcript away, rips it from that duplexed area, and just pulls it out the exit pore. Because it can't physically go inside the RNA polymerase unless it displaces the RNA polymerase, which is pretty hard to do because that's a ternary complex, right? Very hard to dislodge that. So that's my own sort of interpretation of how it acts as a helicase, but it's not really a helicase mechanism if you think about it. And the rest of these pictures here are here to reinforce what I've just said. Hopefully they won't confuse. So I think you have it now straight in your head, so you might not want to look at these pictures. But just look at them if they make you happy. If they confuse you, don't stop looking at them. Okay. Yeah, that looks kind of cool. Yep. Not confused so far. Yeah, that's all really cool. Okay, summary of termination. Both factor independent and dependent mechanisms require RNA polymerase and the transcript to fall out of the transcription bubble. In other words, you have to destabilize, either mechanically yanking it out the exit pore, or destabilize by pausing at a region that's has thermal instability. Rho-dependent termination requires a hairpin, but not at the rut site. I think that's true. It needs to be after the rut site. Bacterial mRNA, though, they're probably coming into something that's going to hurt your head. The exam question, because there's another aspect of this. You know, it's simple as it is now. Why can't we leave well enough alone? Well, it's this situation where you have a Rho-dependent terminator embedded as a cryptic terminator, cryptic meaning silent, a cryptic terminator inside of a coding region. That led some of the early bacterial geneticists to come up with a term called polarity. They found that if they introduced mutations into certain operons, not all operons, but certain ones, they would find that if the mutation was in the first coding region, you would end up not making the product to the first coding region, as you would expect. It has a stop codon. But you would find that you would not have even any RNA for all the coding regions that followed. None of those upstream would be affected. It's just everything after where this mutation occurred. And the mutation was simply introduction of a stop codon. Most operons didn't work that way, but when it did work, because it had direction, it was called polarity. You've introduced a polarity mutant. You stop expression not only of enzyme A, but B, C, and D also. That's normally not the case. If you had a polycistronic message and you put a stop codon in protein or gene A, you would only stop production of gene A, because the ribosome can independently bind to those other perfectly good coding regions. So how is that explained? And that's what you're going to tell me on exam 3. Except I'm going to tell you first, so that you can get it right and have a happy life and be physicians, because I'm going to be needing one pretty soon. This is all just about me, right? Of course, you know what cistron and polycistronic means. So, polycistronic, let's get some basic definitions and understanding going here. This is a polycistronic mRNA, meaning it has more than one cistron. A cistron is, if you don't want to be too clear about your communication, instead of saying a coding region, you say cistron, and people's eyebrows raise and go, oh, a cistron, yes, I knew that. A polycistronic cistron, oh, that's amazing, yeah. It's just a fancy way of saying multiple coding regions. Now, if it's a bacterial gene, the ATG is important, but you can have ATGs all inside the coding region, right? Methionines occur naturally inside of proteins. So certainly you have to have the first ATG because all proteins start with methionine. But in a eukaryote, that would be enough. It would go to the 5' end and just scan along until it got to the first ATG. But you can't do that in a polycistronic configuration because then you'd only translate the first one. You have to have this second component that says start here for translation, and that's the Shine-Delgarno, based on two researchers, I think either Shine or Delgarno. One was the graduate student and one was the PI. It was named after him. Another name for it that's more general is ribosome binding site. It's within 4 to 6 nucleotides upstream of the ATG that should be used to start translation. Okay, so if you're making the RNA, as we can see here, you've got Shine-Delgarno, you got multiple Shine-Delgarnos or ribosome binding sites, each corresponding to the beginning of one of these embedded coding regions. And then you have a transcriptional stop down here at the 3' end of the message, and that can be row dependent or intrinsic. Could be either one. Okay. Termination can utilize intrinsic or factor dependent down here. Termination. Or termination can happen right in the middle of a coding region in some select examples. It provides a way to regulate expression at the transcriptional level of everything beyond that cryptic terminator, and these are all factor dependent. Polarity, and this is what I just explained to you, occurs if at least one row dependent termination site is located between the point where translation is blocked and the ribosome binding site of the next cistron. Row needs to have access to the rut site on the mRNA, and it will in turn cause a termination of transcription in all the following genes. This doesn't mean a lot till you see the picture. So this is the normal AUG or ATG, depending on whether it's RNA or DNA. So this is the RNA since I wrote AUG. The ribosome just binds randomly, just hops around like a little bunny, and it does a Shine-Delgarno here, and then it translates along till it gets to a stop codon. Normally stop codons are at the end of the coding region, and there's the natural one there, but sometimes mutations are introduced deliberately or by accident where you'll get a stop codon put in here accidentally right in the middle. This is stop for translation, right? So the ribosome will bind, but it will come off wherever there's a stop codon, if there's an accidental one here. So this is a case where we're setting up the conditions for a polarity mutant. Because in this case, notice, you have a rut site and a hairpin. These are the two things you need to have a row-dependent terminator. This thing is normally silent or cryptic because the wild-type gene does not have a stop codon right here, and so the ribosome's continually zooming past this site, continually blocking the ability of the helicase row to bind here. So this is a case where the ribosomes are detouring early. They're falling off early, and it leaves this whole region here totally free of ribosomes. The perfect scenario for Rho now to be able to bind this message at the rut site and then cause early termination. An early stop codon, meaning before the rut site, causes the ribosomes to take the off-ramp early, leaving this free. Now you can imagine scenarios on an exam, what I would do. I've got to come up with 3 wrong answers, right? This would be something that would cause termination, but if I put the AUG there, what would happen? Would it terminate? No. The ribosome would come off here, but notice it continually went past the rut site, so Rho would never have the chance to ever bind there, right? So you would, if I had a stop codon right here, this coding region would be no good, but the ones that came after would be perfectly fine in terms of transcription. You would not stop transcription. If the stop codon was put after the rut site, again, the ribosomes would continue to go over rut, so the row-dependent termination mechanism would never kick in. It's only when the stop codon is put upstream of the rut that it can cause a detour of the ribosome, leaving this now permanently exposed to binding by Rho. So it's not a hard complex, but I try to make it that way. And here you see, under normal conditions, you'd have a rut site, a hairpin, but there would be no transcriptional termination here because the ribosome would continually, here you see ribosome traffic, continually blocking and zooming over the rut site, not giving Rho a chance to bind. Transcription of B gene is not halted if ribosome binds at the second Chan-Dargarno, right? So this is, that arrow should be, we should put a stop codon right here. That is TAG, that's a stop codon, it should be right here. If the stop codon is there, the ribosomes will exit early. You will get a transcription to be not halted if ribosome binds at second Chan-Dargarno. But well, I'm not sure what I did with this slide, but it looks all jumbled and confused, doesn't it? Of course it would. Okay, this is, why did I put a stop codon? Let's just assume that this slide got inserted by some mischievous demon that shouldn't be there. Didn't really illuminate. In order for the polarity effect to occur, what conditions must exist? Rut site must be in the last cistron. Nope, rut site must not be in the last cistron. Rut site must be downstream of the last cistron. Cistron, no. Rut site must be in the last, must not be in the last cistron. I think it's 2, right? If you had this set up so that you introduced a stop mutation in the last cistron of a series, there's not any polarity. There's nothing past it to stop. You would get termination occurring, but it's not polarity by definition because you only have blocked expression of the codon that actually has the stop mutation. You wouldn't see the ones that come after it being blocked, because there are none that come after it. That's just something simple I've tried to make hard. And let's see whether I got it right. I could have confused myself here. Whew, I got that right. I'm so tricky. This is like, why did this get here? This is like the exam question. You'll have a diagram that looks like this, and I'll have different places I could put a mutation that results in introduction of a stop codon. So you know wherever I put it, that particular gene is going to be wrecked, not expressed. The question I'll ask you is, gene D expressed or not? That's polarity, right? You put a stop codon here and suddenly gene D can't work? Because transcription has stopped early and you never even make the message for B, C, or D. So these are the possible places I could put it. If I put a stop codon right there, that means the ribosome will bind and come off early. That means the rut site is always exposed. That means transcription will stop and you won't even get the rest of it transcribed. If I put the stop codon here, you're just going to inactivate A because the rut site is continually having ribosome traffic. Rho will never get a chance to bind. Same for if I put a stop codon here. Rho is continually covered by ribosome traffic, so Rho never kicks in. The only thing to happen is you've inactivated cistron A. So there's only one position where the stop codon is going to affect transcription of everything else, where you're going to see polarity happen. It's not that complicated. Now that I've given it to you, all the wrong answers and all the right answers. The only thing I haven't given you is this choice 1, 2, or 3, or 4. And that'll be scrambled for each of you. So this is in the notes, you can look at it. That's the end of termination. That's termination of termination. Are there any questions on anything I've covered regarding termination? Okay, let's move to the next one. How long do we have? 13 minutes. This will be the fastest I've ever covered the operon. The operon, part 1. First of all, we're going to detour with the alpha domain of the polymerase, because as we go into operon, you're going to see that the regulation often involves proteins that either activate or repress transcription. And so to understand the mechanism of how operons are regulated, you need to know a little bit about the enzyme and especially this alpha subunit. You know about sigma. Let's look a little more at alpha, because this is where some of these regulators, specifically CAP, is going to interact. So remember the alpha subunit. There are 2 copies in the core, and each has a carboxy terminal domain that binds upstream of the core promoter. And you can have proteins that interact directly with the C-terminal domains of the alpha subunit. And in some cases they activate, in some cases they repress. Mostly they activate. And they activate by making protein-protein contact with the CTD of alpha. What would you expect that to do in terms of promoter strength? Would it increase it or decrease it? Increase, right? It's another point of contact, another sticky contact that's going to make sure that the polymerase doesn't float free. It's a very simple mechanism. Protein-protein contact. Another way that alpha can make the promoter stronger is by direct interaction with the DNA sequence here. If that's a strong interaction with the UP element, then that will also keep the core polymerase from floating away. That simple mechanism is contributes to most of the regulation of transcription. Interactions with the CTD of these two alpha subunits. So let's look at the different configurations that can occur in, but that'll be a little bit later. This is the strongest promoter on the planet. The bacterial ribosomal cistron. This makes the structural RNAs for the ribosome. So you need a lot of these things. And this is the it's polycistronic, but it doesn't encode a protein. This is what the promoter looks like. It has the start of transcription, a -10, a -35. And then it has a UP element, and this is where the two carboxy-terminal domains of alpha bind. This particular promoter has other positive acting transcription factors which further increase the activity of this promoter, and we'll not talk about those. FIS is the name of the factor. You really don't need to know that. Just look at the contributions here. This UP element increases the activity of this promoter by 60-fold. You get another 5-fold increase. So it's about a 300-fold increase together of having these other elements of the promoter outside of the so-called core promoter contribute to activity. These are some ancient pictures of how this might work. This is actually a flexible linker. It's about 19 amino acids long. The up element, you need to know where it is. It's from -40, so it's just 5 base pairs upstream of the -35 site, right? 40 to 60, it's located lots of places, but that's a good place to start. How does it interact with DNA? First, we're going to talk about how it interacts with DNA, the UP element, and then we'll go into how it interacts with protein activators like CAP. What you'll see here is it has a series of helices and turns. This resembles helix, turn, helix, resembles, if you squint, a helix turn helix, which is just about the only way that bacterial proteins recognize DNA elements. Sigma is an exception in domain 2.4, but the CTD of alpha interacts with configurations of helices that look like helix turn helix, except it's not. This is called helix hairpin helix. Helix, hairpin, helix. Not helix turn helix, but helix hairpin helix. And it does not bind the major groove. It binds the minor groove, which is exceptionally minor or narrow because it's a stretch of A's. This is unusual. I will point out 3 instances in this course where a protein recognizes DNA through the minor groove. This is one example, the first example. So I'd love to ask this on exam. The domain is helix hairpin helix. Helix hairpin helix. And this illustrates what I just said. This is the DNA's footprint. You see where it does bind. It binds from about -40 to -60. Here's some more examples. Many promoters, proximal. They used to think that because A sort of cause a curvature of DNA that maybe that was contributing to its ability to activate genes. Now we know it is simply the strength of the contact with the U element. That's a take home there. Of course, there's room for 2 of these CTDs to bind right next to each other. So you have a proximal, which means close, and distal, far. 2 sites here that occupy the domain between -40 and -60. Both are very AT rich. Now you see some different numbers here, 170-fold and 340-fold. I would not ask you how much it stimulates. Different people, depending on how you do the experiment, you get different numbers on the contribution of the UP element. UP elements often increase recruitment of the polymerase to the promoter, except when it doesn't. How could it not enhance? Too much of a good thing. If it's a perfect UP element, the CTD of the alpha goes, oh man, I found you, baby. I'm never going to let you go. And the polymerase can't get unstuck. It's like it's tethered. Like my mother, she bought me a duck in the spring one year when I was a small lad. It was not such a good idea in hindsight. I lived out in the countryside, and the duck was always in my mother's flower bed. And so my father, being not a farmer, decided, he'd read about farmers and bulls and stuff, that perhaps that we should tether the duck. And so the duck had a big rope around his foot in the backyard until the sheep, my mother gave it to the lady selling strawberries, you know, in the truck with the scale that's swinging back and forth. She's shaking the bell, strawberries, strawberries. My mother runs out, how about duck? No, you can keep the strawberries. But she didn't tell me for years later, and it scarred me for life. Okay, that's a digression. So in some cases, it can be negative because it tethers. These are the different configurations that the alpha CTD can make contact. Can be on one side or both sides or totally on the other side. The catabolite activator protein, or CAP, is the predominant example of proteins that activate transcription by making contact with the alpha CTD. This is an experiment to test the mechanism. You know, there were lots of early theories that AT richness enhanced melting the promoter, some people would say. Others said it was the curvature imparted on the duplex by the AT richness that imparted the strength of the promoter. And others simply said, no, it's just how sticky it is. It just sticks the polymerase onto the promoter with greater affinity. And so to prove that, they said, instead of having a UP site here, we're going to take the lambda, which is a virus, you know, the phage, repressor, and since the lambda repressor forms a dimer with itself, it has affinity for itself, we're going to take this dimerization domain from the lambda repressor, and we're going to cut off the CTD and make the alpha subunit have the lambda repressor dimerization domain. So now, if you're binding lambda repressor where UP should be, there's a chance that the polymerase is going to get stuck because of its affinity for the dimerization domain, and that should activate transcription. That certainly has nothing to do to a natural configuration of anything, but it's simply to prove the point that the sequence here makes no difference at all, because this is the repressor binding site, no AT richness here. But it does provide an opportunity for the alpha subunit to make contact with something to cause the polymerase now to have greater affinity for the promoter. And this actually stimulates transcription. If you do the experiment a little differently, you put the DNA binding domains in this place, then the alpha subunit is going to bind to the repressor site with very high affinity, and it represses. So this explains both of the possibilities, that it can activate and repress, and the common theme is simply manipulating the affinity of the alpha subunit, which is a part of the core, to to the promoter. How much time? We're out of time? Okay. We'll leave it there, and remember to start looking at some of the earlier, last year's lectures on these topics, and I will go as far as I can next time.